Well, good morning, and welcome to a session on clinical trials, room for improvement. My name's John Marshall. I'm privileged to co-chair the session with Ricardo Ferreira from Spain, and we have one fewer talk than was originally advertised. The first speaker, who is going to be speaking on systematic reviews and meta-analyses, has had to cancel. And that means we'll have more time for questions. I was just saying to a couple of the speakers, that means that each of you is allowed to ask 2 questions after each presentation. But we'll start the morning session then with Kathy Rowan from the United Kingdom. Kathy is the director of ICNARC in the United Kingdom, and she's going to speak to us on randomized clinical trials versus observational studies. Kathy.
Thank you. I hope it's okay if I take off my mask to speak. Yep, good. Okay, lovely. So yes, RCTs versus observational studies. So obviously, it's a very sort of polarized, combative sort of situation. In the blue corner, we've got RCTs, and in the red corner, observational studies. So let's just get to the crunch of the big difference between randomized clinical trials and observational studies comparing treatments. And essentially, it comes down to the magic or the power of randomization. And without randomization, you do not get an unbiased estimate of treatment effects. So what is randomization? We all sort of know what it is, but it's essentially the process of assigning participants by chance to groups that receive different treatments. Each participant has a chance of being assigned to either or any of the treatment groups. That's the key. And successful randomization, now remember, randomization isn't like a sort of label that means every study that uses the word randomization is a good study. Always look at the randomization, but successful randomization requires that group assignment cannot be determined in advance, can't be predicted in advance. The person doing the randomization doesn't know which group, which treatment group that patient will be in. So let's look at randomized trials. Let's look at the observational studies now. So what's the sort of the equivalent of randomization in observational studies? Well, essentially, it's receipt. So patients are allocated to treatments essentially by receipt. What do I mean by receipt? It's the process of assigning participants by choice to groups that receive different treatments. That's similar. But each participant is deliberately assigned to either or any of the treatment groups. So the person doing the determination of the treatment that the patient will get actually determines which treatment group they go into. And receipt, therefore, means that group assignment can be predicted or determined in advance. You know that you're going to put that person on that drug or give them that intervention. So going back to the comparison, I've tried to use the same language here. Randomization avoids bias by distributing the characteristics of patients that may influence outcome randomly between treatment groups, so that any difference in outcome can be explained only by the treatment. So it's great. Receipt, or determination, or just comparing patients who got X with patients who got Y, invites bias, as the characteristics of patients that may influence outcome, now these might be unmeasured or unknown between the treatment groups, may differ, even if you carefully match, because you can only carefully match on the things that you know about, the things you've measured and the things you know impact on outcome. So that means that any difference that you then see could be explained by these unmeasured or unknown factors, as well as by the treatment. So you haven't got this pure treatment effect when you're looking at observational studies. So does that mean observational studies, the sort of RCTs, right hook, are out for the count, really no value whatsoever? Well, I'd like to kind of maybe just sort of qualify that. So there's back to our sort of, randomization sort of schema here. So you have a sample population that you randomize to treatment A or treatment B. I'd just like to focus on that, first of all, that sample population that we randomize. So if you go to any randomized trial, you'll see a long list of inclusion criteria and a long list of exclusion criteria. And what that sort of often does is take the sample population away from the population of interest. So we might not include kids, we might not include people over a certain age, we might not include pregnant women, we might not include people who are determined clinically. We may even allow equipoise to come in. So there are trials that say, if you're really sure you want to give them A or B, don't randomize them. And what you're doing in that situation is taking the sample population away from the population of interest. So you end up with that question, is the sample population actually representative in the trial of the whole population? Observational studies, when you do an observational study, none of that really. You describe the whole population. You compare the treatments in the whole population. And so probably what I would argue is what we need to do is harness the strength of both. We did a trial a long time ago looking at the management of patients with pulmonary artery catheters, and we got an effect size. And then there's a technique that you can use. We did some methodological work where if you look on the left here with the table, you've got the patients in the trial in the column 1 who didn't receive a pulmonary artery catheter, patients in the middle column who did, and then you've got a non-randomized study running alongside to just look in real practice who was getting them and who wasn't. So these are patients just in observational day-to-day clinical practice. And then you can compare the characteristics of the patients who got the pulmonary artery catheter in the trial with those in the usual real world. And then through statistical techniques, you can actually take the RCT estimates, sort of adjust for how they differ from the population, and then, if you like, get some sense of what the treatment effect might be in the real world, in the real population. It's a bit like adjusting for the strangeness, perhaps, of the trial population. And this was sort of done by clever people, clever statisticians that we worked with. But it's just sort of thinking about taking the sample average treatment effect and, if you like, translating it into what might be the true population effect. So the other thing about trials is they are a sample. They're always done on a sample. And we know the sample size is the number required to answer the main trial question, and it's calculated using a power calculation. What we normally are meant to do is have sufficient power to detect the minimal important difference in the primary outcome between the groups. What we all tend to do is go, oh my goodness, that looks like rather a large number of patients to randomize, and we'll never get that trial done. What if we look for a slightly bigger effect size, and that will be slightly less patients? So we often do what I call the minimal fundable difference. And often we do trials, me included, hold my hands up, that are insufficiently powered. We're looking for too big an effect size. Back to observational data, describes the whole population and describes multiple populations and multiple outcomes. So you've got all the data in observational studies. And so again, why don't we harness the strength of both designs, use our observational data to help establish the optimal primary outcome, use our observational data to help inform the minimal important difference, and then obviously, as we do, use our observational data to inform the power calculation. Without that, without the observational data, I think the trials suffer. Treatments and interventions in trials. I think we rather provide what we might call ideal rather than real-world treatments. You know, we might have algorithms or protocols, and we check that everybody's adhering to them, and we call things protocol violations and all kinds of things like that. But that sort of goes on in the real world. You know, not everybody gets what they're supposed to get, or gets all of it, or gets all the dose, or gets it every day, or gets it at the right time of day. And what we do know is in trials, the control group or the usual care group always do better than what normal people do in real life practice and care. So we know the sort of situation is artificial, often called the Hawthorne effect. And what we know in observational data is you get the real world treatments, the real world interventions, and you get it in the real world context. So again, I would promote harnessing the strength of both. So our observational data can help inform delivery of treatments that is closer and more relevant to real clinical practice. We can use our data, our observational data, to understand true delivery in practice. And then everybody who knows me will know I'm a big fan of what you might call effectiveness rather than efficacy, i.e. that pragmatic RCTs rather than explanatory RCTs, they're designed to evaluate the effectiveness of interventions closely as possible, mimicking real-life routine practice conditions, so that you've got a better chance of understanding what the effect size would be back there in real life. So going back to our trial schematic, I would argue, and I know others would agree with me here, we often do our RCTs a little prematurely. We might not understand all that we need to understand before we sort of rush in and do the trial. I'll hold my hand up. I've been there too. And I would promote to us that the initial evaluation that we do, it's not hypothesis testing, but it's hypothesis generating, should probably be an evaluation within observational data to then inform what you might call the sort of ideal evaluation, the unbiased evaluation in trials. And again, harness the strength of both. Let's generate and inform our hypotheses from observational data. Let's inform the mechanisms of treatments. How many times do we jump in when we truly don't know the mechanism of the treatment on the outcome before we do the trial? And as I've already said, observational data inform the power and the sample size and how the interventions that we're going to evaluate in the trial are delivered in real life. No two ways about it, whether you are US dollars or euros or Brexit land, pounds, randomized controlled trials are really expensive to do. And let's be honest, real life out there, just collecting the observational data, much, much cheaper to conduct. And again, you're probably getting the sense of where I'm coming from on this. It's about harnessing the strength of both. This is Paul Young's trial, the peptic trial. It was efficient design. It was based on databases, ICU databases. There was no additional data collection in Australia and the UK, so very data-enabled. 26,000 or nearly 27,000 patients across 50 ICUs, across 5 countries, predominantly 2, Australia and UK. And as Paul likes to tell us, it cost $18.5 US per patient. That's quite cheap. So data-enabled and efficient design is all about embedding in observational clinical data. So again, harnessing the strength of both. And then lastly, I think it's about reflecting on the fact that sometimes you can't randomize. There are situations, they may be ethical, but often they're much more to do with trying to evaluate what I consider the most important things in care, which is the service delivery and organization of care. And it is just a couple of our studies. You can't randomize people, say, I wonder if chucking people out at night is good or bad for patients. I'll tell you what, we'll do a randomized controlled trial and see if people will consent, or families will consent to their loved one being sort of discharged in the middle of the night to see if it's better or worse. Again, when you're looking at strain on ICU, you've got observational data and you want to, so some situations you cannot randomize, and observational data are definitely king in that situation. So the title I was given was RCTs versus observational studies. I would say it's not a case of RCTs versus observational studies, but would like to leave you with a situation where it's actually RCTs and observational studies. That's where the strength in our research lies. Thank you very much.
Thank you. Yes, yeah, we are going to have some minutes for questions, so please remain in the podium. There is any question from the audience?
Kathy, would you comment on, particularly given the situation in the United Kingdom, strategies to make trials simpler and more efficient by integrating them into healthcare?
Yeah, sure, I'm very happy to. So I think one of the things we have to be really careful about is not oversimplifying. So I think Simplifying the platform and the delivery of trials, absolutely fantastic. You want to make them as easy to do. You want them to be as part of clinical practice as you possibly can. But in that, you don't want to simplify the lack of understanding of the mechanism. It's sort of the detail, it's still a kind of a controlled clinical, you're still trying to make sure you've got high internal validity, even though you want the greater external validity by embedding in clinical practice. So we are now doing a larger number of what you might call incrementally growing data-enabled trials. And that might be that essentially, because the data collection could be a huge burden on intensive care units, as far as possible using data linkage, using regular data sources that just exist anyway. We're very lucky in the UK, we have an NHS number. Other countries may not have something similar. This is a unique identifier, and that allows you to link across many different data sets. You've obviously got to be really sure your data you're linking to have the kind of accuracy, completeness, validity, and reliability that you would impose upon your trial and your trial database. But I think it's the way forward alongside, and I know Tony will talk about this, the notion of the adaptive platform, the not recreating the structure every time you want to do a trial, but having a structure that's in place that allows you to do either concurrent or subsequent trials.
You want to ask something?
You sort of touched upon it, and you'll, I guess, touch upon it in your next presentation, but the limitation of an RCT is, accessing the population, and then you made it sound like observational studies would automatically access the entire population, but can you speak to the challenges? Because they may not. You might not have the data, or you might have data that needs to be curated, and so that's not that easy.
No, I oversimplified for 15 minutes. You're absolutely right, Francois. So I think there are a number of countries around the world that have international sort of ICU databases, where they have either complete coverage. So in the UK, we cover England, Wales, and Northern Ireland. And so pretty much, if from our data, if we give out a statistic, you can be pretty sure it is a true statistic for the whole population, or nearly the whole population. There are, as you think you're alluding to, observational studies, but it's the same as trials. Always look at a study. So I sort of said to you, look at randomization. It's not a big tick. If it's a randomized trial, it's suddenly a brilliant thing. It can be very poorly done. The other thing to do is to look, either a trial or an observational study, what were the number of sites? Were the sites all actually predominantly teaching hospitals, or was it the big, well-resourced centres? Because as soon as you move away from the whole population, you're actually beginning to bias the sample that you're looking at. And that does occur in observational studies, which I think is what you're alluding to, Francois. In the UK, we're lucky. We have a really good network of critical care units and a kind of culture for data collection, and that allows us to embed our trials, and we link to many trials.
I want to ask you about the control group. What we have to look in the control group. Sometimes we see that the difference in mortality or in any other outcome is because the outcome in the control group is quite far away from the outcome we have in our patients. So what we have to look in the control group?
So, it's the first thing we need to do about the control group is understand, what did control care look like? So we know all the debates around, is it really usual care? And to understand usual care, you actually need observational data to know what usual care is, type thing. And very often in the kind of rather idealized or controlled situations of trials, we idealize in the control or the usual care type thing. So it's no longer real usual care, it's slightly more shiny and polished usual care. And I think it's just about, one, being sure and understanding what was delivered in the usual care. So you do need to know about co-interventions in the control group as well as in the intervention group. And then I think the other thing is you need the observational data so you can understand, again, the population in the control group. What did they look like? What did the trial inclusion and exclusion criteria do in terms of changing the characteristics of the patients? And then use observational comparisons. And as I say, there are techniques where you can actually understand the biases that the sampling for the trial brought to the population who took part in the trial. But that is where observational data are king, because they allow us to situate our experiment. And every trial is an experiment, and what you're looking for is how the experiment situates in the real world or the wider context.
Thank you very much. So we are going to move to the next presentation, which is by Dr. Melvin Singer from London, UK. And he's going to talk about COVID trials and reality checks.
Thank you very much indeed, Ricard, John. Hi, audience. Kathy set me up beautifully, so it's a lovely talk. And actually, I'll follow on from where she's saying. And my talk is COVID-19 trials and reality checks. Right, so what I'll say will probably please some, upset others, and I'll be, oh, sorry, it's reformatted a bit, but I'm sure I'll get a few bricks thrown at me. Oh, he's just an ivory tower academic, not living in the real world. But to quote a Rolling Stone, I view myself as an unpure purist. But I think hopefully, it's important to have these discussions, have these debates. We have to be pragmatic in a pandemic. So I looked up what's the opposite of pragmatic, and you can apply perhaps one or maybe even all of those opposites to me, but hopefully romantic is one of them. I put their drug companies, sorry, it's not drug company, this is an academic trial, but they can still be done to a standard quality we normally expect of a randomized controlled trial. This was an academic funded trial, began at the beginning of the pandemic, multiple sites, multiple countries. They did matching placebo limbs, and even when the hospital couldn't cope, they came up with a way of trying to blind the treatment. They enrolled in just 2 months over 1,000 patients, and they recorded carefully adverse events. So that is what we come to expect of randomized controlled trials. It can be done. We know one size doesn't fit all. So we've learned this from decades of failed sepsis trials. So why should COVID be any different? Oh, it's a long pneumonitis, but we've learned it's different. If you look at sepsis trials and what we've learned from 40 years of failure, we know sepsis is an umbrella syndrome, and there are multiple clinical and biological phenotypes. Well recognized. There are marked variability in biological responses despite the patients being equally ill. When we look at subsets, be it a priori or post hoc, again, there's some suggestion of benefit and harm, but these are rarely followed up, followed through, quizzed more formally. I think we've learned that we need to be able to be a bit more acute as to how we treat patients, personalized treatments, to identify the right timing to give a drug, the dosing, the duration of intervention, and ideally we need to take blood and other samples to actually inform the science, the biology, and why different groups behave differently. So when we go to COVID, yes, we recognize that COVID is actually quite variable in terms of biological and clinical phenotype, predominantly respiratory, however, clinically. There is a marked variation in biological response. Yes, there are these really interesting subset analyses, but they haven't actually been really queried or challenged, which I think is, as I'll show later, quite disappointing. There's been very little attempt, by and large, to try and personalize treatments, and very rarely have concurrent blood samples been taken to try and inform what's happening. We've also learnt from sepsis trials that we need decent phase 2 studies to inform phase 3 design, to try and get phase 3 design, the big trials, done as well as we can possibly manage. We know 28-day mortality is a poor endpoint. We know open-label trials are open to bias. We know that many patients live or die regardless of the intervention, and because obviously we need convenience to try and get the right number of patients we think we can recruit in the given time, the effect size is often conveniently overestimated. So when it comes to COVID, same issues. There's been a rush to go into phase 3 without actually properly working out what we should be doing. Most of the trials look at 21, 28 days. Open label trials, I'll show you later, are open to bias, and many patients, you'll see often these large phase 3 trials are often conveniently underpowered. English expression, more haste, less speed. You make better progress with a task if you don't try and do it too quickly. And I think this is a retrospective reflection, but actually I was saying so at the time, we need to understand the disease better to then inform how we design the trials. And it was the point Kathy was making about observational trials leading on to better designs of RCTs. We published this in November. I think we submitted it in June 2020, so just 6 months or so after the pandemic started. And this is from clinicaltrials.gov. There were about 150 different targets. Where was the science that informed all of these different targets? It was every drug company, device company, academic group with something in the cupboard or on the shelf. Oh, let's try this. It's anti-inflammatory. Let's do a trial. Hang on a minute. What was the logic? We wouldn't do that in normal life. Why should we do it in COVID? I wrote this recently with Andre Khalil, an editorial, and this is a few months ago. At the time of writing, there were nearly 3,000 interventional trials logged onto the clinicaltrials.gov website. Only 1 in 7 had been completed. And a fifth of these interventional trials are large phase 3 trials, a third of which came from industry, 2/3 academic, investigator, government department initiated. Again, only 1 in 8 had been completed. And how many of these huge number of trials have shown a positive outcome? Very few, and I think even in these trials there are questions. I'll come to that in a few minutes. The other thing that worried me is that we normally have good safety monitoring. That often wasn't done in these pragmatic trials, and that worries me, because we don't know. These drugs were not tested, we rushed in, we didn't have often very good safety monitoring. And where was the underpinning science? So, cytokine storm, lots and lots of publications, cytokine storm. More. This came out last month. Current evidence suggests that immunopathology may be responsible for the pathogenesis. I agree, especially cytokine storm. Hyperproduction of an array of pro-inflammatory cytokines is closely associated with poor prognosis. Is it? There was this nice meta-analysis from Cliff Deutschman and colleagues that came out last year, where they looked at what had been published in terms of, here is interleukin-6. And if you look at the COVID patients, they're barely different from the normal range. In ARDS, hyperinflammatory and sepsis, you've got essentially a couple of log orders higher. And in cytokine release syndrome, you give CAR T therapy, you can get a real cytokine storm. It's way higher still. So it's not a cytokine storm, is it? And when they looked at other reported inflammatory cytokines, again, similar issue there, and also anti-inflammatory cytokines too. Right down the bottom comes COVID. So there was there was an inflammatory response, but certainly it wasn't huge. And we knew this right at the very beginning. This is one of the early papers that came out of Wuhan in The Lancet, so a big journal, and look at what they found. Let's pick interleukin-6 to begin with. Again, most of the values were at the healthy, normal control range. And you can see here, there were a few that were elevated, but only a few of the patients had high levels, and even those levels aren't particularly high. Interleukin-1 beta, again, a bit higher, small levels, a low-level rise, and again, no difference between those who needed or didn't need ICU care. GM-CSF, again, the vast majority of patients, normal levels. Only a few isolated patients were high. TNF, again, a bit higher, but again, no difference between ICU and non-ICU patients. So do we have a cytokine storm, or perhaps more of a gentle breeze? So is there therefore a strong rationale to block cytokines? Maybe in some selected patients, absolutely, but to give it to everyone, I'm not so sure. And sadly, as I mentioned before, the large majority of these trials didn't collect any blood samples to actually inform the science, to understand why some people respond, some people don't, or why some trials are negative, but within that there may be subsets who may have benefited. This is a nice study that came out of Spain. It's an observational study, and 146 patients were studied, and very interesting little bit of data. So they looked at patients where they took the bloods beforehand who had high or low interleukin-6 level. So if you look at the patients who had high interleukin-6 levels, where they didn't give tocilizumab, there was a 36% mortality. In those where they gave tocilizumab, there was a nice improvement in mortality, a reduction in mortality. However, in those patients with low levels of interleukin-6, those who didn't get tocilizumab had a low mortality. Those who got tocilizumab with IL-6 had an increased mortality, so it went in the opposite direction. So we may be benefiting some, harming others. Again, it's not a prospective randomized trial. They did logistic regression, and there was this big difference, this interaction effect. And if you look, the other point, I'll come to 28-day mortality. If you censored your collection at 28 days, you'll see here the high IL-6, didn't get tocilizumab, high mortality, and all of the others were much better. However, if you stretch the trial out, look at the fall-off in the low IL-6 tocilizumab-treated patients. A large number of the mortalities happened after day 30. So I'm going to pick this as an exemplar study. Big trial, talked about tocilizumab, the recovery trial. So it was open label. The logic was, patients who were inflamed, CRP is driven by IL-6, so clearly, high CRP should equate to a high IL-6 level. And I'll talk a little bit in the next slide about the weird dosing regimen and the primary outcome, 28-day mortality on intention to treat. So if you look at the dosing regimen, to my mind, it was a bit weird. How did this arise? So basically, if you were 91 kilograms, you got 800 milligrams. If you were 90 kilograms, you got 600 milligrams. So just being 1 kilogram heavier, according to the estimation, you could have got a 33% higher dose. And you could give, at the clinician's discretion, another dose 12 to 24 hours later. Let's compare this against the prescriptions for cytokine release syndrome, where there's much, much, much higher cytokine levels. Here, it's 4 milligrams per kilo. In the tocilizumab trial, it equates to about 7 to 10 milligrams per kilo. So you're giving double the dose, and here you can repeat it within 24 to 48 hours rather than 12 to 24. So how was this actually arisen? How did this dose get chosen? Don't know. Again, observational trials, a lot of people were giving tocilizumab ad hoc. Again, perhaps that could have informed how we designed these large trials. We know that Spanish study I just showed you highlights it very well. A triumph of modern intensive care is our ability to prolong death. So what happens after day 28? This came out of Germany recently in PLOS One from Christian Karaganidis and colleagues. You can see here 30-day all-cause mortality in patients who were invasively ventilated, 43%, and there was a big step up, an 18% step up, or 20% step up rather, in hospital all-cause mortality. So people carried on dying after the event. And if you look actually at ECMO, it's even bigger, 42% at 30 days, and by the time they left hospital, there was a 50% increase. So it is important to keep following these patients through. Recovery have not reported, even in steroids, in IL-6. Why not? And interestingly, in this German study, you know, a quarter of patients were readmitted, which I thought, wow, gosh, that's huge. Okay, let's go back to tocilizumab. So we had a 31% 28-day mortality, 57% were discharged from hospital, leaving 12% still in hospital. And you can presume these are the sicker end of the spectrum. And likewise, in the control or usual care group, 15%. So what happened to them? Don't know. Did tocilizumab help some people get out of hospital quicker, but maybe potentially harm patients who didn't get out of hospital quickly? We don't know. So what happened to them afterwards? Not reported. And there is a risk of bias with open label studies. Let's go back to tocilizumab. You can see here that of the patients randomized to get tocilizumab, nearly 1 in 5 didn't get the drug. We don't know why. It wasn't recorded. Or if it was recorded, it wasn't reported. And likewise, 3.7% of patients who shouldn't have got it were given it by the clinician. So as Kathy said, yes, it's real life, but wouldn't it be nice to know why? Were these patients who didn't get it getting better? And therefore the clinician thought, oh, they're getting better, I won't give them the drug. So therefore they're not exposed to potentially the harmful effects of the drug because they're getting better. We don't know. Why not? We should do. And again, I'm sure Kathy or other clever trialists can tell me why, but everything's done by intention to treat. There are very few trials that actually report per protocol analyses, even in the supplements. So it would be nice to understand what happened to the patients who actually followed the protocol. Not reported. This meta-analysis came out. John was one of the co-authors. Manu Shankahari, lovely colleague in London, sort of led this work. And overall, the view was that there was an association with a lower 28-day all-cause mortality if you've got an IL-6 blocker. But if you look here, again, lots of interesting things are buried in supplements, and people often, unless you're anally obsessive like me, don't look at supplements. This is really interesting. Look at the impact of, on the top, we've got here a load of blinded placebo-controlled trials, and on the bottom we've got the open-label trials where the clinicians knew what was being given. So in terms of the weight, you know, a quarter of the patients went into the blinded trials, 3/4 into the open-label trials, and clearly recovery was 50% of the whole. So clearly that has quite an impact. But the way the randomization was done, actually a fairly similar number of patients, 3,000 to 3,500, if I can read rightly, sorry, blinking, well, essentially got the active drug in the blinded and the open-label trials. Oopsie daisy. So if you look there, the odds-risk ratio in the open-label trials, 0.8, significant benefit. If you look at all the blinded trials, 0 impact. If anything, a trend in the other direction. Why? But it's interesting, just follow the line down and you can see the open label trials are shifted to the left compared to the blinded trials. Why? Similar paper just came out on anakinra in Lancet Rheumatology. Exactly the same. All of the open label trials, some against historical control, shifted to the left. The only randomized control trial to date, bang in the middle, no effect. If you look at overall, a 4% absolute improvement in mortality, so a number needed to treat of 25, but clearly it was better in the open label trials, but in the blinded trials, as I said, it went in the opposite direction. And finally, subset analyses. I've got lots of other things. I'll just pick on these things, really interesting. So yes, we recognize that interleukin-6 blockers and tocilizumab work better in the less sick patients. So once you got onto a ventilator, it didn't have the mortality outcome effect. Sex, really interesting. Males benefit, but not females. Same with the recovery trial on dexamethasone. No benefit in females. Why? We don't know, but isn't that interesting? Are these drugs sexist? CRP, again, you would have predicted, if CRP is a surrogate biomarker theranostic, that the benefit would be seen in the higher IL-6 CRP levels. But the benefit was seen statistically in the mid-range, and the high levels where there were a lot of patients enrolled, as many if not more, no benefit. Steroids, really interesting effect. So if you got steroids, you did well. If you didn't, it was shifted the other side of the line of unity. That's the overall, and again, this is the recovery trial data. Women bang in the middle, and if you didn't get corticosteroids, no effect. So in summary, I think, I'm not denigrating the fact it was an amazing effort to enroll all of these patients at pace. However, I do worry that we sacrifice quality in many of the lessons we've learned over 40 years of sepsis research, ARDS research, etc., for expediency, pragmatism. And there are lots and lots and lots of remaining questions. So I think we've missed a golden opportunity. Should we have done better? I'll leave it there. Thank you very much indeed.
Thanks very much, Mervyn. I think there'll probably be some discussion on this. I'm going to let Kathy start it.
Well, we want an interesting discussion. I do apologise, I have to leave immediately after too. But I love to take the questions.
Okay, I'm just
Throw the grenades.
getting out my gun. No.
Please.
A lot of your talk was actually based on data that the very trialists have put out. That's the first thing I would sort of say. So without the meta-analyses and the split by open-labeled, and so what I'm sort of saying is to defend the trialists, I think they're aware that they were doing things fast and that there's more to learn, and they've started to actually unpick that and present that and give you the data for your lovely talk. The other thing that I would say is, in the H1N1 pandemic, we didn't randomize a single patient into a trial that published in any way to help us during the H1N1 pandemic. In the COVID-19 pandemic, I think we took, and I think you agree with that, a monumental leap forward. Are the challenges, is your critique valid? Absolutely. Was 28-day outcome chosen because we didn't really care, or was it chosen because when you looked at the survival curve for COVID-19, it was the point at which about 95% of the deaths had occurred, and we could have waited for a year to do the outcome evaluation, but then the pandemic would have been over and we wouldn't have had knowledge on the effect of. So it's just that sort of, whilst I take your challenges on board, I agree the critique is valid, what we were doing is balancing speed, trying to help these patients, trying to help the clinicians caring for these patients against sort of, having to make some decisions around sort of, expediency. Comment, question.
No, you're right. There was this fine balancing act, and so you could understand the need to rush. We need to come out with a treatment. We need to show immediately we've got a drug or an intervention that will work. However, we've learned as a community over the years that doing it the way we've done it in COVID hasn't been effective. Trial after trial after trial has failed. So we've learned to try and get a bit smarter, be it 28-day mortality. And the problem is, yes, a small number of people die, but not the ICU population. And, this population here will know that many of the patients who they had were still in a bed on a ventilator at day 28. So, yes, it will apply to the majority of patients who never go into an intensive care unit, but the ones who did, often they did have a prolonged stay. And as I showed you, the recovery trial, a lot of patients were still in hospital, and you can presume after a month they're the sicker end of the spectrum, not reported. So I think there's this balancing act, and as I said, there are all these subsets. Why don't women benefit? Why? And again, people aren't talking about that. It's not being flagged up why women don't benefit.
But the reason that women didn't benefit,
Yeah.
and the 28-day mortality actually was from the ICU data.
Oh, no,
So I think the reason you know all these things to query is because the trial.
yeah, but the problem is we don't then have the blood samples or whatever to go back and actually try and understand why. So why didn't we collect some biology at the time? Because we learned that from the sepsis trials over the years. Yeah, collect some samples, put them in a biobank, and then you can answer these really interesting post hoc findings. Because I wouldn't have predicted that females would have no response and males would. But now we've lost the opportunity to try and understand why. Tocilizumab, why did it only work with steroids? Why? Again, missed opportunity. Throw bricks at me, Tony.
Following on that, from that, I would say there are samples. That's what ISIRIC-4C, another platform and observational studies, collected samples. They can be. So they did the samples. They can be mixed together. I just want to correct a few important points. The female bit, if you actually carry on down on your table, female, there's a statistically significant improved outcome for the not deteriorating to need ventilation, ECMO, or dying. I think most women would rather not go on ECMO, and so I don't think people should come away thinking these drugs don't work in women. Picking subgroups in the sex that is less likely to be ill, is less likely to die, it's just a statistical thing when you pick out one subgroup. I think they do benefit, and I think it's important people don't go away thinking I shouldn't give these drugs to women, because I think they are having a benefit.
Well, could you show me where? Because
the next
tiny
next
numbers
line
got
down.
ECMO, Tony. Tiny numbers.
Next figure down, I mean,
Yeah.
I can show you the figure here,
Yeah.
but anybody wants to read them, and if you follow it down, the outcome for those three is the confidence interval is below 1. It works in women. I was going to say, the bit about blinded, you mentioned the blinded studies. Those were the studies that also didn't give steroids because they were done early. So again, I think it's really important people get the right message. You need to give these drugs with steroids. You talk about the crossovers. Crossovers, if you get them, are actually a trialist's nightmare because it brings the results together. So the fact that you still see a result, even though not everybody got the drug, suggests the drugs really work. But I agree with you, if you're getting better, you wouldn't give the drug. And maybe I did point out, just wanted to say, because the dose business, I mean, it's not often you'll see me supporting the recovery trial, but I know that they would have chosen the dose because the vials come in 200 milligram vials. There's a practicality. I don't know if you were in the same pandemic as me, Mervyn, but we were discussing which drugs we could put in the same syringe together because we didn't have enough syringes. Therefore, why would we give someone 1 kilogram more, a whole vial of tocilizumab? There is a worldwide shortage of these drugs, so it's a pragmatic thing. It comes in 200s. It's weight-based. We'll do it in bands. This happens all the time in all aspects of medicine. And so these trials have shown us how can we improve care in hospitals, treating the patients we have now. And I'm glad the results came out from recovery before they had even followed everybody to 28, because if we were waiting for 6-month mortality, we'd have to come back in March next year to even see the preliminary results. And thank goodness we've actually got on and treated patients and improved it during the pandemic.
Right. Sure. You've got one person applauding. I would turn it around the other way. You've given me the trialist apologist view. Wait, let me answer. Okay, so you can answer back. So the apologist view. Oh, well, steroids, in the blinded trials, steroids were not given. True, but what about recovery? Large numbers of patients, steroids, if you've got steroids, the effect, no steroids, no effect. So it was replicated in that study. The argument you made about, well, the lack of benefit puts patients closer together. Well, maybe, but as I pointed out in my talk, if you're giving it to patients, you're not giving it to patients who are getting better, that potentially means you're not getting the side effects of a drug if the patients are improving. So again, we don't know. Why weren't they reported the reason for the crossover? We don't know, and that's a failure, and it's a tick box.
Absolutely, we would all love, and every trialist would love to have longer CRFs, but then we'll have...
Yeah, but the trial I showed you at the beginning, the ACT trial on the remdesivir, they did that. So they could do it. Why couldn't the rest of us do it?
But they only did 1,000 patients and...
only 1,000 in 2 months.
But didn't, precisely, and stopped and didn't give us a clear answer. After that, the WHO's differs to other opinions because no one's sure whether remdesivir works or not. Because their trial stopped at 1,000, it may have had loads of information on 1,000 patients, but actually we don't know from that trial whether it really works or doesn't work.
But then the corollary is to then repeat the trial with bigger numbers informed by that data.
Yeah, and that's what happened in Solidarity, and then people
Yeah.
said there's not enough information, but I can see we're...
No,
I'm getting
what-
in maybe we should stop and, I don't know.
This is wonderful. I'm
other
just sort of
people can
thinking that,
John, you can pick who gets the last word.
I was going to say Tony goes next. I was just going to give him an opportunity to catch his breath before he has to get back up on the stage to make the argument. But I just wanted to comment that I recall very early during the pandemic.
And I've got to talk in the next
Very early during the pandemic, a very eminent trialist, an extremely eminent trialist who was working with REMAP-CAP made the comment that we're not ready to start the trial. If we had another year, we would be ready to start the trial. And obviously that just was not the case. So I think, Mervyn, you've made a pretty good case for the progress that's been made based on pragmatism, number one. And number two, for the fact that, as you said at the beginning of your talk, we want to individualize treatment. And the reality is if you're individualizing treatment, you're doing it because you're going to find responses different patients. It's not a shortcoming of the trials. It is simply a validation of the
But
argument.
the problem
you
is
made.
going forward, you know, Tony obviously believes in tocilizumab, I don't. So therefore, when another drug comes along, do you have to give it with tocilizumab or can you randomize against it?
But ultimately, that becomes your responsibility as a clinician, is to use the information. I think the trial's responsibility is to generate it and to challenge the kind of discussions we're having right now. So it's been very good. But thanks very much for a
Thank you.
wonderful, enlightening discussion. Okay, so the next speaker, as it happens, is going to be Professor Tony Gordon from London, who's going to speak to us about what we've learned from adaptive platform trials.
Thank you. So as Mervyn's actually leaving, I can say what I like now, can't I? So I'm going to talk about what we've learnt about adaptive trials. My disclosures and acknowledgements are shown here. And just to be clear, the drugs were provided in REMAP-CAP, but no other industry involvement in the trials. Obviously, more important academic conflict is the UK chief investigator in REMAP-CAP. And I'll try and give you personal opinion of, obviously, why I think they've worked, but also I'll try and point out a few things where we can still do better. I don't think they are the answer to everything, and there's still a lot for us to learn and improve on. So, as you saw from Mervyn, there have been many thousands of trials listed. Many of them haven't started, many of them didn't report, and I would argue that probably, apart from the vaccine trials, which have clearly been extremely successful, the platform trials have been amazingly successful. Here I've listed the outputs from REMAP-CAP and Recovery, 2 platform trials that have all produced results on 9 interventions. The ones in bold are the ones where there's been benefit, but I think we all understand the importance of learning what doesn't work as well. And as to celebrate 40 years of this meeting, I've been coming to them for about 20 years. At most meetings, Mervyn and others will stand up and say, if only we had some trials where we had positive results in critical care, it would be so much better. Now we have them, so I think we should celebrate and move on. There is still an awful lot to learn, and as I said, I don't think we've got all of the answers, but we are making progress. You may have a clever protocol, but I think that's, it's not just about having the right protocols as well. This is recruitment in the UK during the pandemic. And we were, as REMAP-CAP, you know, was set up for a pandemic. We were open in a few sites before it started. And we were trundling along. You see the sites that are open shown in orange. We were trying to get more sites going as we knew the pandemic was coming. But then we got the chief medical officer gave us this urgent public health priority. So all hospitals were told, take part in these trials. And you can see the massive effort we did, opening 140 sites. The team at Imperial and working with Kathy and the team at ICNARC did an amazing job getting 140 ICUs open in a few weeks. You can see the benefit. We did okay in the first wave. We recruited pretty well, but everybody was overwhelmed. But you can see the real benefit of being set up in a platform. When we had our peaks over Christmas, more than 450 patients recruited in a week over the new year. It was a very miserable new year in the UK. And I don't know, I mean, I've done other trials. It's often taken me 2 years to recruit to a 450 patient trial. We did it in 1 week. So it shows the benefit of having a platform, how you really can recruit very well. But you do need that infrastructure as well as a smart protocol. So I think some of the efficiency from platform is just the logistics. If you run a series of trials, you can set them up, close them down, wait to set them up again and go, and you can see it takes a lot of time. I think critical care has got itself better, maybe not even formal platforms, but almost like a network. If you've got a team of people, the collaborative groups, we've been able to run one trial straight after the other and you get results much quicker. But wouldn't it be more efficient if we could actually combine them all together and test them simultaneously in this platform, and then we'll get our results for multiple interventions much quicker. So this gives a scheme of what a platform trial might look like, a review that came out just before the pandemic, in fact. And I would suggest all platform trials are adaptive to some degree. The recovery trial perhaps has less features than REMAP-CAP does about adaptation. So that not all adaptive platform trials have to look the same, but the common feature is you're obviously adding interventions or domains over time, you randomize patients, you generate the results. But importantly, you have these fixed termination rules that determine whether you reached a conclusion. If you haven't, you keep on going and you keep on randomizing. As I said, there are aspects here, and I'll talk about it, randomization weights. It's not compulsory to do this, but there are all various aspects of adaptation you can do. It also doesn't have to be a Bayesian design. REMAP-CAP has used a Bayesian design. You can still be adaptive with a frequentist trial. Recovery is entirely frequentist in its design, but it still adapts. I think the Bayesian allows more adaptation, but just to be everybody's clear, you can do that more with a frequentist trial as well. And I'll give you a good example why I think we need these sort of things. This is a traditional A versus B trial from an excellent group of trialists. You'll know many of them, comparing 12 milligrams of Dex versus 6. Well-conducted trial, and they did exactly what they said they were going to do. They get their final result, and they've got a p-value of the primary outcome of 0.066. Mortality, 0.86 relative risk, confidence intervals crossing 1. It looks encouraging that maybe it works. But of course, they haven't shown that it works, and they've said there's no difference, and of course reported the confidence intervals are wide, i.e. imprecise. We haven't got a definitive answer yet. And I would suggest that if they had been in an adaptive platform, rather than having a fixed termination rule, that they did their best assessment that they could possibly do, they wouldn't have stopped, they would have kept on randomizing, and by now we might have a definitive answer about whether the dose of dexamethasone was better to increase. I think it's also to be important what an adaptive trial is not. People have said to me, oh, it's okay in REMAP-CAP. You can just do what you like. That is not how an adaptive trial works. I've seen many protocols that say we're adaptive, we're going to recruit 500 patients, have a look at the data, and then decide whether we carry on or not. That is not how it works. Everything has to be written down. You don't make it up as you go along. Everything we have is pre-specified. So you have adaptations, you accept that things may change, but you do need to pre-specify them. That has its complications. You obviously have to write this all in a protocol. The way most people do it is having a master protocol, or as we call it, a core protocol that describes the overall structure of the trial. Who are you going to study? How is it going to be run? But then you can have these appendices, as the way we've done it, that describe the interventions as they come in or come out. So you don't have to change the core. And of course, if you're going to be a big international platform, you need to deal with region-specific areas. So we had appendixes for this. So it is all written down. This is great. One of the downsides of this is a huge document that builds over time. And people have said to us, can I just have a look at the protocol so I understand it? And I'm not sure we've got the right summaries right. Tom sits on our DMEC, and at times he said, can you remind me what's going on? There's an awful lot going on here all the time and so on. It is challenging. So I think this is one area we need to improve the summaries so that we can understand everything that forms part of the trial. So this is what it looks like as a scheme and how we started. Got the overall platform, and you meet general criteria. So for COVID, it was that you're an adult patient in hospital with COVID-19. And then we've put each of the interventions. We started with steroids, antivirals, and immune modulation in a domain. These are drugs that work in a similar way and that you would want to randomize to one of the options. And the way to think of it is almost like each domain is like a mini trial itself. You share the same primary outcome, but you can have domain-specific outcomes. So it's more efficient like that. And as you see, it built over time. It became much bigger than I think any of us ever thought. You could test multiple interventions. These are the different domains and the different interventions that we've had open at some stage or still have open in the UK. And so you're testing multiple interventions. I think, and again, this is just my personal opinion, or I should say the most we've had randomized for one patient is 7 in 7 domains. I personally think that's probably a bit too many. I know from feedback both from our research staff and also patients that go, I could handle 4 or 5, so maybe that is the optimal number. But I said it's just a personal opinion. But these are the number of domains we've had open. You don't have to have every intervention open at every site. It may not be available locally. You may not have equipoise and say, I don't want to randomize in one of these domains. And that's fine. Patients may be ineligible. But then a patient gets randomized to multiple interventions, one from each domain for which they're eligible to. This has a number of advantages. One I hadn't really thought about so much was, particularly in a pandemic, where I think patients may say, oh, but there was a tendency we would all want the experimental treatment, particularly in a new disease we're all worried about, and to be in the placebo arm. Well, if you've got multiple interventions, it's very unlikely you're going to be in the placebo or the control group for every single intervention. That may not be important, but I think it makes people feel a little bit better about taking part in research. And similarly for the clinicians, they say, well, I don't know that steroids, tocilizumab work or plasma works, but I think there's a chance. And if you were randomizing and only going in the control arm, people said, well, I agree, I don't know steroids or tocilizumab works, but there's a high chance that you're going to get one of these experimental therapies. And again, I think clinicians felt easier about randomizing to that. And the other thing is you can test for interactions. And I think we accept that in modern medicine, it's not about giving one drug. In critical care, we give lots of drugs. And we need to assess how do they work in combination. This idea that we mustn't ever randomize 2 patients together. And we saw the importance of that, as you've seen from Melvin, the fact that it seems that the IL-6 inhibitors only work if you give the steroids as well. And we saw a little bit of this in REMAP camp. Scott Berry, our statistician, was jumping up and down with excitement when he looked at the interaction statistics to say, he said, I think there's an interaction there. We couldn't prove it at that time, but it's subsequently, by combining all this data, we've seen it very clearly. So again, the importance of being a multifactorial trial. And I'm just going to spend a moment about an optional feature, shall we say, in randomization and in adaptive trials, is the fact that this idea of adaptive randomization. So in a traditional run trial, you randomized in fixed ratios. It doesn't have to be 1-to-1, can be 2-to-1 if you like, but you put the same number of patients in each intervention, the same allocation ratio, so here 1-to-1 for the whole trial. You get to the end, recruit 2,000 patients, 1,000 got each, and then you look at the results, and then from that point on, you can give the patients the best treatment. With adaptive randomization, what you can do is start with the same fixed ratios, start putting patients into each group. At one of these interim adaptive analysis, the computer starts to see the data, and if one treatment is looking better, it can start allocating more patients to that arm. So you don't have to wait for the patients to potentially benefit once the trial finishes. You can be learning while the trial is still going on. It's still blinded, the investigators don't know, but more patients may be treated with the better treatment. So that could be of use to ensure more patients get treatment. It can get you answers more quickly. Not if you've only got 2 interventions. Actually, you get an answer just as quickly with one-to-one randomization. But if you have multiple interventions, if the one treatment is looking more interesting, you can get answers on that quickly. And as Melvin talks about, maybe some of these treatments may be harmful. Equally, they can be dropped from the randomization. So you actually learn and put fewer patients at risk of the treatments that are either not working or could even be harmful. So I think what I've described is undoubtedly an extra layer of complexity compared to a traditional A versus B trial. There are guidelines to help you report this, because there is more that needs to be reported when you do that. So there's an extension to the consult diagrams and so on that you put up, and I just illustrate ours from the IL-6RA manuscript in New England. We describe all the patients who were randomized. You can see them there in the flow diagram. But there are a group of patients here, shown in the purple, who were allocated to other interventions within the domain, who we didn't know the result of at that time. We've shown them in the figure, so you can see them. They actually appear in the overall baseline characteristics, so they're reported. But we didn't know the outcome at the time. We've now subsequently reported them. So that is a bit different, so I thought it was worth explaining. We also have in the platform other patients who weren't randomized in this domain, and I just wanted to clarify, these patients are not control patients. All the comparisons within the platform happen only with patients who are randomized. All the controls are randomized, and all the treatments are random and readily allocated. So these are here. They provide extra estimates around what is the effect of things like age, sex, region in the world that you were recruited to. They don't take part in the comparison, but again, we've reported them. There's a great opportunity for international collaboration. And I just point out this is wonderful. It makes external generalizability possible. On a personal level, I've made new friends and colleagues. One of the downsides is we're all on different time zones. So this is Colin MacArthur's phone at 10 o'clock, nicely for him in the morning. And if you notice, most of the European investigators are only half awake at midnight or 11 p.m. So it is tiring, and it can be hard to coordinate. I think a few final issues just to finish with. Funding is tricky. In theory, this is an open-ended trial. It's perpetual. But you can set end dates for your funders. But many of them will want fixed deliverables, which can be variable, obviously, in a platform. And we have this in the UK. We're very much contracted to deliver. So I think we need to work on that. I'd say not all ethics committees and regulators understand it. We've been very lucky in the UK, but I know many of the countries in Europe have struggled to understand these concepts. So we need to educate them. Melvin mentioned about blinding. This can be done within a platform. There's no reason. It's best done at the domain level. It would be really challenging to provide, though, multiple placebos. That would get very complex. And I'm not sure you always need blinded trials if you really want to know the effectiveness in the real world of interventions, which is, at the end of the day, what we are after. And finally, there are simple things like even the trial registration. We apparently have broken clinicaltrials.gov. We've used up all the slots that you're allowed for your treatment allocation. So as we add new interventions, we're not quite sure how to add that. We're still working with them. So bear with us if you say, oh, but you didn't add it. We don't know how to add it. There is no more space. So there are lots of tiny little things we need to work on. So I think I've shown how their benefit. I think they're ideally suited to tackling disease areas, particularly when you've got multiple treatments and if you want to look at patient subgroups. If you want a simple answer on A versus B, there are other easier ways to do it, I would say. They are complex. You need an experienced team. They certainly need a lot of planning, but I would say that's what you always need in any trial. The coordination, oversight, and I would encourage critical care to set up more, and that we continue to learn and improve them, because it's not the finished article yet. Thank you very much.
Thank you, Tony, for the presentation and for all the work you have done. Congratulations. Any question? Yes.
Ivor Douglas from Denver. As a huge enthusiast and participant, and realizing that without COVID-19, the platform concept in critical care almost certainly would have taken another 10 years to really come to life. Appreciating that there is tremendous power in this approach to quickly cycle through agents would you please address the issue of type 1 error and the risk for abandonment with too rigid a decision rule around inefficacy? So the opportunity that's lost with A versus B trial when you move to a platform with fairly rigorous stopping rules is that you might significantly put at risk a modest treatment effect that would be picked up in a larger trial that's allowed to go to full enrollment with good follow-up. And I do have concern that we may have missed over potentially efficacious agents through rapid cycle.
Yeah, a good question. Deal with the sort of bit about type 1 error. I mean, it depends on what method you use. So if it was a frequentist adaptive trial, you would, like Recovery is said, we'll recruit 4,000 patients before we look at the results. For the Bayesian analysis, this is all done in the pre-trial simulations to define those stopping rules. So this was done by the Berry team. So looking at likely events and so on, they actually run the simulations and can control for that type 1 error. So for us, for 1,000 patients, the type 1 error is less than 2.5%, so smaller than the 5% that most of us talk about in the frequentist lingo. The issue is about whether you stop for futility. I mean, that's almost a pragmatic decision. And again, you run the simulations and work that out. And it was a case of making that decision, particularly in a pandemic. When do you want to stop and just move on? I think that might be different outside of a pandemic when there's less priority to generate the answers quickly. So if there's a small effect, that may be very interesting. But in a pandemic, we actually needed large effects. And so I think we made those decisions. But we may choose, and if it's a cheap, easy, available intervention, you might choose to really keep on going for a longer time before you declare futility. Compared to randomizing people to ECMO, you may say, well, if it's not having a large effect, I want to stop earlier. So you just build those in. You have to make those decisions.
Thank you. Equally as an enthusiast, I'm just an enthusiast, so putting it out there, but I wonder if we are misattributing the research efficiency during this pandemic to the platform and adaptive design. They're great, multiple interventions at the same time, and you can add more interventions quickly, and all that is great, but the research efficiency wasn't that mostly attributed to the volume of patients that were enrolled in a tiny period of time? And wasn't that largely attributed to a very special system where research was embedded into a public, central, national healthcare? Because unless I'm mistaken, when Recovery took 2-3 months to enroll 6,000 patients, they were only enrolling patients mostly for steroids. And when around Christmas, when you were enrolled dominantly in the TOSI with REMAP-CAP, and REMAP-CAP isn't enrolling as fast in other countries, and other platform trials outside the UK aren't enrolling as fast. So are we sort of confounding the effect here with?
That's an important point, and that's why I put up that slide about recruitment. But I would suggest that yes, I think imagine if every country, let's just say Europe, just to keep it simple, but obviously North America as well, had recorded the 4,500 that we have in the UK. I know we had a lot of cases in the UK, but I think you've all had a lot of cases as well. And if you had been able to recruit all of these patients for all of these interventions, we would have got more answers. And so when I've heard people say, oh, but we haven't got answers on this and that, we could have done that. And if the whole world came together, and I think we've said before, sepsis trials, ARDS trials are not big enough. If we actually come together, and this is, Lenny Derde is talking about it in another room, about the collaboration. We need to set this up as international collaboration. So all of our countries can recruit 4,500. We'll have tens of thousands of patients, and we'll get the answers there. So we need both, and that was why I put that slide up, because I think that is important as well. We are lucky. And maybe other countries can learn from the systems that we have set up, because it is about embedding research in our everyday clinical practice. And we've seen clinicians actually do like it. And one of the benefits we've had is people said, what I really liked is we were randomizing one day, and then it became a treatment the next day. they actually enjoyed this part of their job, and I think it would make all of our lives in critical care more enjoyable if we were able to see these advances because we generated the evidence quickly.
Just a short question. What about co-enrollment in classical trials? Are you in favor, or you think that platform trials do it better?
I would be supportive of the co-enrollment because it's the same concept. It needs to be carefully managed. So we still allow co-enrollment in REMAP-CAP and other trials because there are other trials going on. We always look for important interactions, particularly biological, so you don't want drugs interacting, and there are other scientific reasons. So, you know, if one trial is a trial of ventilation and your outcome is ventilation, in the other trial you won't want to co-enroll. I think it can happen, and it needs to be controlled. One aspect is the burden on patients and families when considering that. So there's an element, it's easier in a platform because it's one consent form and information sheet. We've tried to make our information sheets as short as we could. It obviously is by definition long with multiple interventions, but make it modular so people can see the bits they're taking part. But absolutely, and I think we're seeing the importance of why you need to allow co-enrollment, because you may be surprised at the interactions that you see, and that reflects how they work. And I've seen the trials that didn't allow any co-interventions and said, oh, we didn't like steroids. They went, oh my God, steroids have become standard of care. What the hell are we going to do now? And they wish they had allowed steroids earlier on, for instance. So, yeah.
Okay, thank you. So it's time to move to the next presentation. I invite Tom van der Pol from Amsterdam in the Netherlands to talk about predictive and prognostic enrichment in sepsis trials.
Thank you very much, Mr. Chairman. Oh, sorry. Okay, for something completely different now, well, not entirely different, but it's a little bit more how you can enrich your patient population in such a way that the likelihood that you will find a beneficial effect of a treatment is higher. This is a very simplified scheme of the host response to sepsis. Within the upper part, pro-inflammatory reactions, and in the lower part, anti-inflammatory reactions. And many of these host response aberrations have been targeted in clinical trials in sepsis, and these were invariably in normal, we'll say, conventional trials, so a one-on-one randomization and looking at the effects, so no platform trials. And this is a not complete list of interventions, and you probably are well aware of that none of these interventions showed any benefit. And you might ask the question whether these trials, these interventions, were really on target, because what these trials did, they used prognostic enrichment rather than enriching the population for a certain biological phenotype that might provide more likelihood that these interventions would work in this population. Prognostic enrichment means that you enrich the population for the clinical endpoint of a trial, and in sepsis this is mostly mortality. So this is a very nice scheme from an article written by Hector Wong and Dr. Stansky. And here you see the prognostic enrichment. And for clinical trials, it means that you divide patients with a low mortality risk and a high mortality risk. If you don't have enough patients with a high mortality risk in a sepsis trial, you're very unlikely to show an effect on mortality because the event rate simply is much too low for this. And patients with low mortality risk, you might argue that you can just treat with standard of care. And now comes the next concept, and that's predictive enrichment, which means that you try amongst the patients that have a high mortality risk to stratify patients into those that are more likely to benefit from a certain biological intervention versus those that are unlikely to benefit from a certain biological intervention. When discussing tackling the heterogeneity of sepsis, several concepts have emerged, and I highlight two of them here, which have been widely published now in literature. and that's the concept of the subphenotype and the concept of an endotype. So a subphenotype more relies on clinical features, and it's a group characterized by a set of features that are not necessarily linked by a common pathophysiological mechanism. But still there's evidence that subphenotyping of sepsis can inform treatment selection to improve predictive enrichment. And then there's an endotype, which is defined as a biological subtype, which is defined by distinct pathophysiological mechanisms. And I'm going to show some examples of these. So probably the largest paper that's out there that was subphenotyping sepsis patients is written by Chris Seymour from Pittsburgh. And he used 29 variables that are readily available in each ER, clinical and routine laboratory variables. And he used those 29 variables obtained within 6 hours of presentation to the ER, and he used k-means clustering to find 4 clinical phenotypes, alpha, beta, gamma, delta, and the distribution in these different phenotypes are here. So the most frequent phenotype is the alpha phenotype, followed by the beta and gamma, and then the delta phenotype. So in this scored diagram here, the ribbons between the phenotypes connect to organ dysfunction that's displayed here. So you might appreciate that if you look at cardiovascular dysfunction, this links to the gamma phenotype and also to the delta phenotype. And the same is true for pulmonary dysfunction. And you might also see that the hepatic organ dysfunction is mainly linked to the delta phenotype. So these phenotypes tell you also something about the profile of organ dysfunction in the ICU. They also convey a prognostic, they have prognostic implications. So on the left-hand side, you see the discovery cohort, and on the right-hand side, the validation cohort. The distribution of these phenotypes were roughly similar in these two cohorts, but what you might appreciate is that in both cohorts, the delta phenotype had the worst outcome. So this is cumulative mortality. Now, how can these clinical subphenotypes inform patient selection for trials? And this was done in a very elegant way. So this is data from the PROCESS study that was published now 7 years ago in New England. And this is about early goal-directed therapy, which means that in the first 6 hours or so after presentation to the hospital, there's very aggressive monitoring and associated aggressive therapies directed at ventilation and circulation. And this was compared against conventional treatment. And overall, there was no benefit at all in this trial. I'm not going to go into details, but what Chris Seymour did in this JAMA paper, he looked at the different phenotypes that he could distract from this overall patient population, and then he found that in particular the delta phenotype that was harmed by early goal-directed therapy. Now, you could use this information for trial design if you would stratify these patients based on their phenotype. So this is a Monte Carlo simulation of this same trial. So this is the real trial with the frequencies of the different phenotypes. And you can then sort of simulate the trial with playing around with the frequency of the phenotypes that are present in the trial. So if you would enlarge the population for alpha subphenotype, there would be more patients that would benefit from this treatment. And if you would stratify in the opposite way, so you allow more delta phenotypes in the trial, then you would have more harm in this trial. So you could see that by playing around with the frequency of these subphenotypes in a clinical trial, you can inform patient selection and you can increase the population that has benefit from this intervention, but you can also exclude patients that can be harmed by this intervention. Now let's look now a little bit at the bottom part, so the endotyping, so patients grouping based on a more common pathophysiological mechanism. And this is a study we did a while ago in the Mars Consortium, which was a collaboration between Amsterdam UMC and Utrecht UMC. And the first author, Brandon Skikluna, did all these analyses. So what we did here is we looked at over 300 sepsis patients, and we had blood leukocyte transcriptomes on admission. and there was a huge amount of differentially expressed genes between sepsis patients and healthy controls. And then we just asked the computer, can you tell us whether there's subtypes in this 300 patients that are more alike based on their gene expression profile? So it's a totally unbiased analysis. And sure enough, this is k-means clustering. There were 4 phenotypes, which we named Mars1 to Mars4. And here you see the heat maps. They clearly distinguished between each other. This had prognostic implications. That is, Mars1 phenotype was associated with a worse 28-day mortality, and this was confirmed in 2 independent cohorts. Now, this also has pathophysiological meanings, because if you look at how these Mars endotypes, which were generated in a totally unbiased way, again stressing, there was also different pathophysiological meanings to these endotypes, which is illustrated here. So the Mars1 endotype, which has the worst prognosis, was associated with enhanced heme biosynthesis. We're not totally sure what this means, but this is what we find. And there was basically a total shutdown of both innate and adaptive immunity, as illustrated by the gene expression profiles within this endotype. And the most favorable endotype, Mars3, was actually associated with the relative upregulation when compared to the other endotypes of adaptive immunity. There's also another study that's widely cited. This is coming from the group of Julian Knight, looking at community-acquired pneumonia with sepsis, so all admitted to the ICU. And he used exactly the same approach, hierarchical clustering, unsupervised, of those patients with blood leukocyte transcriptomes obtained shortly after admission. And he identified 2 endotypes, sepsis response signature 1 and sepsis response signature 2. And when he looked more closely at these endotypes, he found that SRS1 was associated with immune suppression, at least that was shown by the gene expression profiles, which pointed at immune suppression in particular in this endotype. Now, is this important? And this is a very nice example how unbiased endotyping can inform treatment selection. This is a reanalysis of the VANISH trial, in which, amongst others, hydrocortisone was tested against placebo. So hydrocortisone is the red line here. And when Dr. Antcliffe and co-workers looked at the effect of hydrocortisone in these different endotypes, they found that the immune-competent endotype was associated with harm if you treat them with hydrocortisone. Now, this is very important information. There's a lot of hydrocortisone trial data out there, and it's somewhat confusing because some trials report benefit, others do not, and it might well be that a subgroup of patients is really benefiting from this, and this is partly obscured by the fact that we also enroll patients that are harmed, such as shown in this slide here. There have been some attempts in recent sepsis trials to do predictive enrichment. So this is the trial published last year, or now 2 years ago, in the JAMA with soluble thrombomodulin, which is an anticoagulant that also has anti-inflammatory activities. And this is an example of combined prognostic and predictive enrichment, because they did enroll patients with cardiovascular or respiratory failure, but also with sepsis-associated coagulopathy, which was defined as depicted here. So they tried to enrich the population for those who had coagulation disorders, because the drug that was given is targeting coagulation abnormalities. The trial was negative, so that's another failed sepsis trial, but at least they did an attempt for predictive enrichment. Then there's also trials in the immunostimulant area, so there's a lot of evidence that sepsis is associated with immune suppression, and there's many interventions have been tested now almost invariably in very small trials, and this is one intervention, interleukin-7, which is increasing lymphocyte counts and also lymphocyte functions. So what these investigators did, they prognostically enriched their population by only allowing in patients with septic shock, but they also predictively enriched their population by only allowing in patients with lymphocytopenia. And sure enough, you can see that these IL-7 interventions then increase lymphocyte counts, so it really does what it's supposed to do. Much attention recently has been given to the so-called macrophage activation-like syndrome as a means by which we could enrich trial populations that might benefit, with patients that might benefit from anti-inflammatory interventions. And this whole macrophage activation-like syndrome interest was triggered in particular by a retrospective analysis of a very old trial with interleukin-1 receptor antagonist, which shows no benefit at all. This trial was done in the 90s, when some of the audience weren't even born yet, I guess. There was no benefit whatsoever. But when they retrospectively analyzed this trial, they found features of macrophage activation syndrome, which was defined as hepatobiliary dysfunction and disseminated intravascular coagulation. And if they only looked at these patients, it was clear that interleukin-1 receptor antagonist, which is an anti-inflammatory agent, had a clear beneficial effect in this subpopulation. So this led to the assumption that if you enrich the population for patients that have the feature of a macrophage activation-like syndrome, anti-inflammatory therapies might work. And then subsequent literature, which I'm not going to discuss here, showed that a ferritin level above a certain threshold might identify patients with a certain with what's now called a macrophage activation-like syndrome. In the same way, you could identify patients that might benefit from immune stimulant therapy by looking at certain markers of immunosuppression, and the most common marker besides lymphocytopenia is reduced monocyte HLA-DR expression. So that's a common feature used in many different trials, which can be relatively easily assessed in the clinic to identify patients with immune suppression. And this is now used, it has been used in a recent small trial, and the results are not known yet, where Mihai and Evangelos, the PIs of this trial, stratified patients with sepsis to either IL-1RA if they would fulfill the macrophage activation-like syndrome criteria based on ferritin levels, or to interferon gamma as an immune stimulant for those patients that qualify for immune suppression based on monocytes HLA-DR expression. And this is now part of a large European trial. Some of the investigators are in the audience. It's ImmunoCEP, it's a European consortium between 9 countries where patients will be randomized to either IL-1RA or interferon gamma based on their immune profile. Now this is my final slide, concluding slide. What I've tried to show you is that prognostic and predictive enrichment can be used to stratify patients with sepsis in more homogeneous subgroups with more similar clinical outcomes, that's prognostic enrichment, and more similar pathobiological mechanisms that are underlying their disease, and that is predictive enrichment. And probably the combination of both prognostic and predictive enrichment is required to increase the success in the field of sepsis, maybe in combination with platform trial designs rather than conventional trial designs. And implementation of these enrichment strategies, you might agree upon that you need bedside testing for that, because the information, whether a patient falls into a certain pathobiological category, you need this information quickly and preferably at the bedside. Thank you very much for your attention.
Thanks very much, Tom, for that very clear and enlightening talk. We have time for a question or two. Let me perhaps just start. I'm curious. I mean, I love this this concept. I'm curious as to the extent to which there's transportability across the different endotypes that have been identified. In other words, how do Mars criteria compare with those of the SRS1, SRS2, and Carolyn Calfee's criteria?
Yeah, so there's 3 groups out there. There's another group that has published based on genomic data, endotyping. And there are similarities and dissimilarities, but some of these endotypes show strong overlap. And I think what's needed, just like in the cancer field, is there's going to be a consensus about this, and then decide upon what the endotypes really are. So you could also, so this is all unbiased, right? So for patient selection, you might also argue we don't need unbiased analyses of these data, but we want profiles that link with the target drug. So, for example, PD-L1 inhibition, you might argue that you want to select patients with abnormal PD-L1 signaling. And can you distract that from, for example, genomic data? I think it should be possible. And based thereon, you could maybe find a signature that's targeted rather than coming from an unbiased analysis.
Tony?
Tom, could I ask you, what do you think about blood as the sample that we measure? And so, going back to Mervin's talk, you showed quite clearly we've seen the IL-6 levels are nowhere near as high as what we've called, and I know Carolyn doesn't like it being called the hypoinflammatory phenotype, but it's a disease that clearly is inflammatory, and anti-cytokines have been proven to work. But it would look like there wouldn't be a subphenotype that you would want to give an anti-inflammatory drug to because of what you measured in the blood. But what's happening in the lung is very different.
Yeah.
Do we risk by missing or getting it wrong by only looking at the blood?
That's an excellent question. So, and that relates to what is an optimal biomarker. So a biomarker, not just one biomarker, but a set of biomarkers, if you look at an endotype, should be obtainable from a body fluid that is easily obtainable. So then you come with blood, you come with urine, and maybe lavage fluid in an intensive care unit. That's about it, I guess. So what you then need is that you need biomarkers that can be measured in one of these body fluids that is reflective of a body situation. So what you need is large observational studies that look at these body fluids, look at biomarkers therein, and combine that with more invasive measurements, maybe in organs, maybe combine it with autopsy studies if patients die, and you can link them to fluid measurements that were obtained while still alive. So the biomarker field is only based on these body fluids that are easily obtainable. In the end, when you want to use them, that's the way to go, I guess. But you would need to link them to more detailed information, and that for, well, I think all biomarkers, none of the biomarkers is available. It's not so easy to do, but it's interesting.
Okay, thanks very much again for a great talk. So we're moving back to the more pragmatically clinical focus. Kathy Rowan's going to speak again on the need for national audits and benchmarking.
Hello again, thank you for staying. I think I'm going to give a different talk. So I was given the title, We Need Audits and Benchmarking. And I suppose I wear two hats in the UK. I'm a clinical researcher, but I also believe in clinical audit or benchmarking. And the reason being is actually they're all trying to achieve the same thing, which is we're trying to improve care and outcomes. And I hope I will give you some information that a dual approach might be an important way to go. So we need national audits and benchmarking. So the first reason I think we need national audits and benchmarking, we call these things different things in different countries, but I'm sure you understand what I'm talking about. I think for accountability. And the reason I say that is for a lot of areas of healthcare, you could go and choose your provider. You go and choose your surgeon. They've done lots of whatever it is you're going to have done, and volume outcome relationships exist, and you think that's the great surgeon who's done lots of those, and I'm going to go and make sure they do my operation for me or whatever. I would put to you that people don't, on the whole, tend to select their ICU. You don't have some sense of becoming critically ill on the whole, and therefore you probably want a system where there's benchmarking going on. So whichever intensive care unit in the UK you end up in, small or large, district general hospital or teaching hospital, you've got some sense that the outcomes for that unit are being benchmarked. And when we do benchmarking, in this case, I've just put up a funnel plot, Windsorized funnel plot, so allowing for sort of over-distribution, and looking at, in this case, the ratio of observed to expected, in this case, acute hospital deaths. And you need a absolutely accurate risk prediction model. It needs to be up to date, but you kind of want to know that you're going to go to a critical care unit somewhere in the middle of the distribution there. And you probably want to be looking at the units that lie 95%, 99%, so where their expected deaths are higher than their observed deaths are higher than the expected deaths. So it's sort of just this notion that the system is getting the same outcomes across the board. I think also we need national audits and benchmarking for comparison. It sounds sort of sensible. We benchmark on a number of potential quality indicators, and I think not everything is zero, if that makes sense. So the delivery of care, the service delivery and organization of care is such that some events will occur. What you want to be sure of is that the event rate in your particular delivery and organization of critical care is sort of within the norm, if that makes sense. So critical care in the UK has recently realized, and actually recently realized over time, that we have a low provision of critical care. And we were getting, if you look at that top bar there, a large number of high-risk sepsis admissions. These are patients who have substantive organ dysfunction before they even get to the critical care unit. That's probably too late to be treating them. And so actually benchmarking something like that and understanding your rate in the context of other places' rates actually puts you in a powerful position to negotiate for more resources. And certainly benchmarking data in the UK have been used to increase resources both locally, regionally, and nationally. I would argue that we need national audits and benchmarking for improvement, so not just accountability, but also for improvement. And this is actually looking at if you apply an accurate risk prediction model from the year 2000 across the years, that what you can see here is the observed outcomes relative to the expected outcomes for a model based in the year 2000 improve over time. So there is some notion that the sort of the nudge technology, so the quarterly feedback, does actually lead to improvements in outcome. But you sort of don't leave it there. What we do is essentially move the goalposts on a regular basis. So as critical care unit outcomes improve, we set the sort of bar higher in terms of recalibrating the risk prediction model, now saying, actually, the expected outcomes we want now are better than those we were expecting before, and we then apply those models. And what that does, if you imagine all the green blobs here are intensive care units and their observed and expected outcomes, you can see as they improve, the green, they're starting to go below the point, things are looking good. You recalibrate or reset the system and you bring them back up to a more even distribution around 1. And this allows for improvement over time. And if you like, the intention is to draw the system to increased nudged improvement. So I've just talked to you about high-risk sepsis admissions from the ward. This may not look like a big improvement, but basically nudging that sort of service delivery and organization. Over time, you see the number and proportion, and each patient who gets to the critical care unit too late actually is someone whose care is actually going to be a greater challenge and a good outcome harder to achieve. What does it sort of mean? Well, in 2009-10, this was the scatter for high-risk sepsis admissions from the ward. And then I've overlaid here what it looks like later. And you can see not only a drop in the overall rate, but actually most places are improving in terms of the service delivery and organization of care around early recognition of sick patients and getting those patients into the critical care unit. Here's another one, unit-acquired infections in blood, starting not a high rate, but 4%, but over time, with nudge technology, with quarterly feedback, bringing those rates down. So clearly, lots of units with 0 unit-acquired infection in blood, but some with higher rates, and again, overlaying more recent data, you can see a general improvement overall, but also for each individual critical care unit. We also need national audits and benchmarking, I believe, for managing policy making and planning. And it's really sort of come to the fore. We regularly share data with our national policymakers, our regional policymakers, and locally with the management in hospital. But it also allowed us, Tony alluded to the great recruitment into REMAP CAP. We were the method centre for REMAP CAP in the UK. And as you can see, we were already capturing those COVID-19 cases. So we were sort of on the case, and that actually contributed also to sort of the network. But we were basically providing, quite literally, data to our Prime Minister and the Cabinet Office on a daily basis in terms, and I think having the network for benchmarking and improvement allows you to have the platform for informing management policy and planning. This again allowed us also to try and be alert to increasing provision over time. So for obviously extending critical care unit provision within a pandemic, here you see the relationship between COVID and non-COVID workload over time. For a data-ready network, so I mentioned REMAP CAP, I think one of the things that is really good about harnessing what I would call national clinical audit and national clinical research is that we're in the situation now in the UK where over 90% of UK critical care units will participate in multi-centre research studies, and that those research studies are embedded in the data collecting network. And I think it's a lovely sort of symbiosis between what I would call nudged improvement via benchmarking and clinical audit, and then also trying to evaluate new technologies or new ways of organising and delivering care. For non-randomised comparisons, again, I touched on that in my previous talk, there are many situations where we're not able to evaluate in a randomised controlled trial a particular way of organising and delivering care. Or, if I go back to the H1N1 pandemic, we didn't get trials up and running, so I think we took a monumental leap forward in the COVID-19 pandemic, but we did have some data where we could try and explore, using sophisticated matching methods, whether ECMO had been a beneficial intervention for patients with the severe 2009 influenza. Hypothesis generating, not hypothesis testing, but I think having those data allows us to start that journey towards trial evaluation. For informing and supporting RCTs, again, I covered that in my previous talk, but if we just take something very recently published, the REST trial, we will have informed the sample size calculation for the REST trial, such that the control group baseline from which the sample size was calculated, we estimated to be 41%, and in the trial, the control group mortality rate was 41%. So it allows us to be more sophisticated in our sort of estimates for our power calculations. And then, because we have the routine data collection for the clinical audit, we actually link those data for the REST trial, and that allows the two to be combined. And we work with all the investigators in critical care trials across the UK to have that kind of mutually beneficial arrangement whereby the data that are being used for the benchmarking and clinical audit are also captured and validated to a standard as you would have in your clinical trial so that they can be linked for trials. And then finally, for nesting RCT samples. And what I mean there is, and I touched on it on my other talk, when you do a trial, you do a trial on a sample population. It's always important to try to understand who went into that trial, and then that allows you over time or statistically to understand the population in whom an intervention may or may not be effective. So it's this notion of understanding the sample relative to the whole population. And then the last thing is we do all these trials, and we've seen lovely examples over the years of even the investigators who take part in trials, who show the intervention is effective, and then looking at their practice downstream 12, 18 months later, and those interventions not actually being delivered in practice. So the research not translating into practice. And I would argue that clinical audit and benchmarking allows you to undertake those activities in a routine-ish way. Here, just showing you one. This is the, on the left, the TTM trial, and the results that were published in the New England Journal in 2013. And then what we did was look at the practice in patients similar to those in the TTM trial. And what you can see is in the 4 years leading up to the publication of the trial results, a sort of concept of moves towards sort of using cooling. And then almost after the presentation of the results, you can see a quick click change in practice as sort of cooling sort of seems to go out of fashion. And obviously, we'll be looking again with the recent publication of the TTM2 trial. So it allows us to sort of try and understand the impact of trial results. And then the other thing is, again, we've heard about the recovery trial and the REMAP-CAP trial. Again, during the pandemic, we were actually monitoring the translation of the use of steroids and the use of tocilizumab and sarilumab in patients after the sort of presentation of those sort of results into the public domain, and the notion that, and we were doing this for our policymakers and our planners. They wanted to see that the monies that they'd put into the research for identification of the effective treatments, that was then being translated into delivery of care to patients. So, in conclusion, I think we need national audits and benchmarking for exactly the same concept that we need clinical research. We're all about improving care and outcomes, and I think it's an important platform and partner to our research, and I think the two are stronger than either one alone. Thank you very much.
Any question? Yes?
Markus Krivars, Helsinki, Finland. Thank you. Very nice talk. So I got two questions. So if you have lots of ICUs in the UK and you look at, say, outcome from severe sepsis, then of course you're going to have variability just by chance. So how do you then determine if something is abnormal based on poor quality of care? Is it purely a statistical question if you're outside a 95% confidence interval, or what is it? Is there any data on what is a relevant difference that's probably related to things like in care?
So we don't take a statistical approach to it. I mean, obviously the comparisons are statistical, the funnel plots are statistical, but in terms of the improvement practices, actually individual places like to look for any of the statistics that we provide, the delivery of care. It's a bit like sort of units doing their mortality and morbidity reviews. So you can either go through all the cases you saw in the last quarter, or you could say, I'm going to work down from those patients who had the highest, the lowest risk of dying, but died. And so you're sort of tailoring and you're trying to learn from those sort of patient care experiences. So I think there is an improvement science, and I'm not an improvement scientist, but certainly the data are picked up by those who are into improvement science with a view. Do we identify absolute differences in care that lead to outcomes? Probably mainly around service delivery and organization. So things around delayed admission and premature discharge are much more likely to come out through these techniques than sort of the content and context of care.
And the other question is can you somehow quantify things that happen outside the ICU? So, of course, if you're in an ICU where you tend to admit everybody, you admit the severe sepsis in the 90-year-old just because you can provide palliative care and you can call in the family and you buy some time, but it's not going to you're not going to do everything for that patient. Whereas the unit that just goes and says, not for sepsis, called palliative care team. And of course, these things will enormously influence the outcome then when you end up in the ICU. I mean, an Apache score works in an ICU, but if you go to a nursing home, it's probably not going to work at all. And you still have people who die with very low Apache scores and very low SOFA scores just because they're elderly and chronically ill.
Yeah, no, I mean, it's a very, very good point. As you say, you're looking in a very sort of blinkered view. We try and look before and after within that sort of blinkered sort of population. I think there is a great push to want to do what you might call whole healthcare audit, you know, to follow the whole patient trajectory. I think the future of that is actually going to be much more within the electronic health record, which will obviously be a whole hospital record. And I also think it will be the future of it is within data linkage. So it is quite interesting that even within the COVID-19 pandemic, we've actually been leaking primary care data, nursing home data, to hospital data, to ICU data, to post-intensive care data. And I think that is the future, but I think, you know, it's a sort of working with what you can work with. It's sort of a pragmatic solution, but it doesn't tell the whole story.
Thank you very much. We are running a little bit behind the schedule, so we are going to close this discussion here. Thank you very much, Kathy. And we are moving to Gordon Rubenfeld from Toronto, Canada. He's going to discuss about the pandemic change. The pandemic changed the way we use evidence in critical care.
Thanks. I think I have 4 minutes, so good
No,
luck
no,
to
no,
me.
we're good till 10:15 at the very
Okay,
latest,
so they're not going to close the doors. Well, thanks for staying, which is great because this is actually a 50-minute talk that was turned into a 15-minute talk that will now be a 4-minute talk. So the assignment was about how whether the pandemic has changed the way we've used evidence. And believe it or not, I'm going to try to argue that it hasn't. I'm going to spend a little bit of time criticizing a website, which maybe sounds bad. I don't know how many of you are aware of a website titled the Frontline COVID Critical Care Alliance. Have people heard about this website of some critical care clinicians who are making recommendations, including ivermectin and things like this? Those of you who are on social media, I suspect, will be aware of it. And the reason why I think this talk is usually a talk that's meant to actually explore evidence-based guidelines, right? Like surviving sepsis guidelines, the ATS guidelines on mechanical ventilation. And the purpose of the talk is not to tell you to use low tidal volumes or to tell you to use ivermectin or tocilizumab or anything. It's meant to actually let you look, for those of you who haven't been on guideline panels, to kind of look behind the door about what's really going on. And try to explain what's happening. And the reason is that all three of these, this Frontline COVID Critical Care Alliance, the Surviving Sepsis Campaign Guidelines on COVID or sepsis, and even this publication from probably more than a decade ago now, the Ventilatory Management of Acute Respiratory Distress Syndrome by John Marini and Luciano Gattinoni, two of our greatest minds in mechanical ventilation. These are all guidelines. They're all meant to be evidence-based guidelines. They're guidelines to tell us how to take better care of our patients. And I'm going to argue to you that they're all different. And the reason they're different is actually incredibly important. So for this website that I'm actually going to talk a little bit about, it was created by 4 intensive care doctors who were all pretty prominent doctors, very intelligent. 1 or 2 of them are pretty distinguished researchers. And they have evidence-based recommendations on their website. And for pre-hospital, they have recommendations about vitamin D and quercetin and melatonin and zinc. And for hospitalized patients, they have recommendations about steroids and ascorbic acid. Interestingly enough, tocilizumab doesn't make their list. Anti-androgen therapy and even therapeutic plasma exchange. I gotta tell you, I think I'm an okay doctor. I don't use any of these for COVID patients. And I'll have more to say about whether, if you do or not, what that might mean about you. This is how I think most people think. I guess I should say by means of disclosure, at least intellectually, I've sat on the Surviving Sepsis Campaign guidelines. I've sat on mechanical ventilation guidelines for ARDS. I'm a guideline guy. I like them. But they don't work like this. This is not how they work. The way they work isn't you sit down, do a systematic review, do a meta-analysis, and say, okay, here's where we have very good, strong evidence with randomized trials. This is what we'll tell doctors to do. And here's a place where we don't have any evidence, so we'll say don't do it. That's not how they work. I wish it was this simple of reading the papers and deciding how strong the evidence is. This is a framework I came up with actually now many years ago after the first guideline body I was on that I found very frustrating. It actually looks a lot more like this, by the way, which looks a lot like what you do at the bedside. Right? We all have priors about different treatments and whether we think they work anyway or not. We read papers, and then we end up with a belief. I have a strong belief that low tidal volume mechanical ventilation is helpful for ARDS, because I had a strong prior belief about ventilator-induced lung injury, and I read some randomized trials, so I really believe it strongly. But even if you think a treatment works strongly, you have to take into account lots of other decisions before you decide to use it, right? Is it expensive? Is it safe? Does the patient want it? There are many physician values involved that I'll talk about if we have time. And then if you're writing a guideline and not taking care of an individual patient, the guideline writers have conflicts of interest. And we've gotten very worried about this when it comes with pharmaceutical conflicts of interest, but we all have conflicts of interest, right? For my whole life, I've given lectures that you should use steroids for everything. I have a kind of a conflict of interest maybe about steroids. But after you account for all of these things, that's what goes into the strength of the recommendation. So you've got the strength of the evidence that's informed by priors, and then you have the strength of the recommendation that's informed by lots and lots of other stuff. And you say, well, wait a minute. I thought this was supposed to be this objective process where smart people are sitting down and making objective recommendations. What's all this other stuff? Where's this coming from? And you're exactly right. All of those factors come from the guideline writers' internal beliefs and feelings and attitudes. I'm going to talk very briefly about priors. I usually spend a lot of time on this, but I'm going to actually skip this slide about UFOs because it will just confuse you, although it's fun. Anybody believe in UFOs? All right, that has a lot to do with priors, by the way. Turns out if you believe in aliens, if you have priors about aliens, you believe in UFOs. But the reason I'm skipping that slide is because 5 years ago, and when I talked about Bayesian analyses and Bayes' theorem, people just kind of went to sleep. And I think now we're beginning to understand this theory. We have this nice reanalysis of the Eolia trial by Ewan Gallagher and colleagues that basically shows across a strong range of prior beliefs. And by prior beliefs, I mean, if inside, psychologically, you've always thought ECMO was a really good thing, if you look at the Eolia trial, you think it's a really good thing. But it turns out, even if you're pretty skeptical, even if you're me, and those of you who know me know I'm pretty skeptical about everything, even if you're me, and you're pretty skeptical, after reading and understanding the results of the Eolia trial, you should have a pretty strong belief. So you have a good idea of why priors, namely your beliefs before you confront the data, have a big effect on our beliefs. Now that bothers a lot of people because science is supposed to be objective, and when you start talking about beliefs in psychology, it gets very confusing. Sorry. That's the way statistics work. And I'm not going to go through reanalyses of other trials. So you might say, well, where do these priors come from? Like, do I inherit them? I don't know. They're psychological, really, in a lot of ways. People have tried to study them more objectively. But in fact, they come from the universe of things that you know, from all the basic science literature you know, from the pathophysiology you know, from what your annoying consultant told you when you were a junior doctor goes into that. This is where priors come from, and they are a bit psychological. So the problem then is that different people have different priors, and they do. I will tell you that most of the pro-con debates that you see at meetings are not debates about the science, they're really debates about priors, because the science is the science, and what people end up debating are their priors. So it's very distressing. I'm sorry it's such a short talk because I can work you through it psychologically and give you some therapy in a longer talk. It's very distressing to find out that all this science is actually pretty psychological, but it is. Fine. I'd like to talk a little bit about grade, because one of the big differences between the website and the consensus of 2 and the surviving sepsis is methodology. So I'm not saying it's perfect. I'm not a grade acolyte, believer, but grade gives us a methodology for doing guidelines, and we never had it. And it's very detailed. It's a bit like a Bible. And I will just tell you, because a lot of people feel this, guidelines do not solely rely on randomized trials. It is not the case that guidelines only use randomized trials. Many guidelines and many recommendations are based on observational data. Many are based on no data, like, should we use antibiotics for infection, right? Something one of our chairmen is a bit skeptical about, reasonably so, when you look at the evidence. So guidelines do use all kinds of evidence, but they just use it carefully. And one of the problems is that guidelines may not weigh, for example, pathophysics. They call this indirect evidence, the grade gurus. They may not weigh the indirect evidence from surrogate studies, from animal studies. You may weigh it very heavily. Tom Vanderpoel may look at those studies and understand them much better than me, and may weigh the indirect evidence much more strongly than me. He's probably right, but the guideline bodies as a consensus panel, they'll look at that data, but they may not weigh it as heavily as you do as an individual doctor. Fine. So those are priors. Sorry, a quick run through priors. What's all this other stuff about cost and risk and values? In fact, guideline bodies do include all of these, or at least try to. I will tell you one of the things that bothers me the most about guidelines, although they are slowly changing, you'll find statements about values and risks, is that guideline bodies tend to handle this part of my model the most informally. Because it's pretty hard to say, I think you should use high-dose famotidine for COVID, because really it's incredibly safe, and I have some values about doing something. As a doctor or as a guideline body, I feel we should do something. And this drug is pretty safe. So the weighing there is not so much the weighing of the evidence or priors, it's really weighing values, the value of doing something during a pandemic, where you feel like your hands are tied, right? And this drug, I think, is very, very safe. That's a values decision. All right. So I think this is pretty close to the end. Evidence-based guidelines, the ones that you're used to reading, the really, really well-done ones, like surviving sepsis or the ATS mechanical ventilation guidelines, they're not, even when well done, based solely on a systematic review and meta-analysis, right? They incorporate priors, and they incorporate their priors, not your priors. They incorporate the priors of this consensus panel. Now, their priors may be better than yours. That's the hope, right? But they also include the guideline panel's subjective weighting of a lot of other things. And remember, they're doing it for an average patient. You're doing it at the bedside for an individual patient, and they're doing it for an average. And the hope is that a bunch of really smart people with expertise from lots of different areas will weigh, on average, these risks and benefits and costs better than you do. We do it every day. Most of the evidence that we have, these are a list of things that we do, some of you do, I do, based on pretty moderate or no evidence. And we do it every day in the ICU. And when you do it, and if you're an academic physician and you're teaching, you do what I do. I tell the training doctors that this is an evidence-free recommendation, and I'm basing it based on my priors and my experience and my assessment of the risks and benefits in this individual patient. You do it every day. The guideline body does it as a group for an average patient. And hopefully they do it better than you do. They probably do it better than me. So, and we make this, by the way, this is where, and there are lots of them, there are lots of them in all evidence-based guidelines. There are always recommendations that are strong recommendations with weak evidence. It comes up in surviving sepsis, it's in the cardiology guidelines. Every guideline body makes strong recommendations based on weak evidence. And you might say, well, how is that possible? And the reason it's possible is because guideline bodies are not simply a synthesis of the weighting of the evidence. And when you incorporate other things like priors and assessment of risk and cost, you can come up with strong recommendations with weak evidence. So I'm just going to conclude, because we really are running late. My comment about what the pandemic has done, and I apologize for going through these slides like this. My comment about what the pandemic has done to evidence is nothing. The pandemic hasn't changed the way we use evidence. But what it has done, just like it's illuminated the cracks in our social infrastructure, just like the pandemic has illuminated the cracks in our medical provision of care, just like it's illuminated the cracks, at least in the United States, in our political environment, the pandemic has illuminated the cracks in the way we use evidence. Because the fact of the matter is, the website that recommends quercetin and plasma exchange, that is making strong recommendations with no evidence or weak evidence, it really is doing a kind of guideline. Because whether they say it or not, what they're doing is making strong recommendations with little or no evidence, in some place modest evidence. It's not wrong. It's not bad. What's bad is their methodology, because they have no methodology. But the idea of strong recommendations with weak evidence is throughout medicine. And what's bad and what's dangerous about what they've done, and I'll say it dangerous, is that there's no methodology and no disclosure. And I'll just finish with this, for example. For example, if a doctor gets up at a meeting like this and says to all of you, I've reviewed the meta-analysis on ivermectin, even taking out the withdrawn fraudulent study, which there was, and even acknowledging the fact that all of the papers, almost none of these trials have been peer-reviewed. They're all on preprint servers, and they have many problems. Even acknowledging all of that, I use it. And the reason I use, I don't, by the way, I'm saying theoretically. A doctor could say, I use ivermectin because there's some evidence, and it's very scary, and I think we should have a drug to use, and it looks like it's safe. If you make a recommendation like this with all of those caveats, I don't have a big problem with that. You disclose your methodology and the values that are driving it. When you make a website and you make a strong recommendation with no caveats like that, I think it's unethical, and really it's not great medicine. So that's my conclusion. Thanks very much.
Well, thanks very, very much, Gordon, for kind of discussing the sausage-making aspects of guidelines and exposing its underbelly. We do actually still have time. Our session, we've got another 5 minutes.
I ran through like crazy for you.
Well, but I'm going to start with Kathy, and then my co-chair has a question as well.
So, Gordon.
I'm going to need some water for this one. Yes, Dr. Rowan, Professor Rowan.
So what you've made me reflect is actually we should be very concerned. You sort of fairly glossed over what I might call the constituency of guideline panels, okay?
Yes.
They're experts, they're this, they're that. They're usually male and white, but that's just another thing entirely.
No, it's not another thing when it comes to this.
And there is actually, and then the second thing is how discussion is had and whether there are loud voices in the room and less loud voices in the room, etc. And there is science about both the constituency of a panel and whether it is representative and reflective of all the sort of caregivers in the process. And then there is also science around how you achieve consensus through discussion and evidence and that kind of type stuff. So it's just really maybe asking you to reflect on, all the science seems to be on the bit about the evidence and the priors but actually, should we be thinking a little bit more about...
No, but thank you for letting me add a new section to the talk. So what Kathy's pointing out is that, and in fact, and Kathy also knows this because she's been on a lot of these panels too, consensus should be in quotes, right? It's usually the person who's the most persistent, right, as opposed to the consensus view. And also, by the way, for those of you who get grumpy about the all-male panel stuff, hopefully after this talk you'll realize just how important a diverse panel is, right? Because I've just told you how important all this stuff about values and preferences and priors. of the people on the panel are. So don't get grumpy about diversity in these panels, because the panels are basically incorporating a lot of who they are into the recommendations.
Richard.
Yes, I have a question. It's a technical question regarding the guidelines, but I think it's something that is interesting for the discussion. What about the PICO question? Because I think in the PICO question, which is the beginning of the process, there is high risk of introducing priors there that is going to drive all the process later.
Well, I might understand that I think the PICO framework, right? This is the patient intervention, and sort of the way you frame the question is a good framework. But perhaps what you mean is just what treatments you decide to ask the question about. Of course. Yeah, of course it does. Yeah. Which things you decide are on the menu to put on there to discuss drives everything. Absolutely.
And the consensus is not there. Usually this is, the consensus is when you evaluate the evidence, but there is not a lot of consensus when you
I don't know.
develop.
It's a really good question. I'm actually not so smart about where the actual, like which things you decide to ask about begins. But you're right, that isn't part of the, not always part of the consensus process that I've been in. But the consensus is around the strength of the recommendation, right? And yeah.
Okay, any last final questions?
Anybody else? Head spinning. Sad to find out how subjective statistics is. It's
Francois.
Scary, huh? Yes, sir.
I'm so completely on board with the priors, but I just wanted to mention maybe.
You want to talk about UFOs?
No. I think I wanted to highlight the grade methodology.
Yes.
As you've mentioned, or guidelines methodology in general is evolving.
Yes.
It's really evolving. The grade gurus are not blind or unreceptive to the criticisms that were quite fair, and there has been a huge, and so you've mentioned the balance of panels, which you like intrinsically, that's why it's important. The notion, someone else mentioned, like the heuristics of when we're faced with a problem, clinicians, even guideline creators used to say, oh, there's no evidence. All we have is a small number of observational bias studies. But the trend is in that direction. That's the best. Well, no, the best evidence, the best estimate of that evidence is that it's unreliable. And the only place where I would perhaps, I found pretty recently, and particularly during this pandemic, a slight change in that due to this rapidly evolving methodology, we've seen occurrences. And perhaps it was because of the pressure, the global pressure of the pandemic and the global burden of huge expensive medicine, and there's been a little bit more accountability of guideline makers in saying, I'm sorry, the evidence is just not sufficient, and taking into consideration all of the non-evidence-based factors, but we have to make a recommendation against until further, and like the case with ivermectin. And so despite the trends, despite everything else, we'd like to, but we'll have to, so a little bit more accountability perhaps to say.
I think that's true. So first of all, I agree that the evidence is, the grade methodology is evolving. I have some opinions that we don't have the time for about how it's evolving and how pragmatic it is in its application, but it's way better than nothing. It is evolving, recommendations evolve. Everybody, particularly during a pandemic, the evidence is evolving. It will always be a problem to take weak evidence, whether it's observational data or biologic plausibility, when it comes to very safe and inexpensive repurposed drugs. It will always be a problem about making a strong recommendation or not, because even grade groups will make strong, because I've been on them.
It should happen less and less.
It
should not actually happen.
Yeah, well, we'll see. Because there's certainly evidence that it continues to happen in cardiology. Thank you, Chairman.
Okay, thanks so much. Thank you. Thanks to all the speakers for a really stimulating session this morning. Thanks to the audience for being part of that stimulation.